So you might ask, why would I want to watch a grade 10 student explain grade 9 and grade 10 biology? And that's because, one, we're the same age. I understand what you're learning, and I understand what actually really matters at this age. And I also understand how some people don't want to just watch a whole video and they think that that's boring. So this video is made not to be watched, but to be listened to instead. So if you have some way, just try and turn off your phone, just listen to my talking as I explain everything very like over audio much more than I do visual, and I don't have any visual diagrams put up, so don't worry about missing anything. It's just all audio. So have fun listening to this. Let's talk a bit about info on the cell cycle, and I'll be going into things like DNA, RNA, and all those different things, and this will just help you out a bit in biology. So I also have this really cool thing that I made, and if you want this, you can totally grab it. It's just in the description below, and I think it looks really cool with all the colorful designs. So this is kind of like a little just cheat sheet. Right? So let's first talk about the info in the cell cycle. So 90% of the time in the cell is spent in this phase called interphase. Interphase is not just its own phase, it includes four other phases: G0, G1, S phase, and G2 phase. So that is four phases in total. G0 is kind of this resting phase. So in the other G phases, you're growing, right? I mean, the cell is growing and stuff is changing, proteins are being added in. In the resting phase, it kind of just sits there. And little things will be done, little repairs of DNA can be done there, but it's not really where things actually grow and proceed. G0 is not a phase that all things go through, and it's kind of a resting phase if something makes a mistake, but a lot of things do stay in G0. Some cells like nerve and muscle cells stay in G0 indefinitely. Next is G1. So Gs are often thought of as growth things, growth phases, but it's actually gap phases. So G1 is gap 1 phase. So the cell grows in size, produces proteins and organelles, and ensures it has enough resources for DNA replication. It also checks for damage before proceeding. And this is how all the phases go. There's these little checkpoints that make sure, hey, there's no issues, and then they'll proceed to the next point. Next is S phase. This is synthesis. The cells duplicate its entire DNA, creating two identical copies for future daughter cells. So eventually, the cell will split apart into two cells, and in this phase, it creates the stuff that lets you split apart into two cells. So the cell duplicates its entire DNA, creating two identical copies for future daughter cells, and histone proteins are also introduced to help package the DNA. So next phase is G2. This cell, at this point, the cell continues growing and produces proteins needed for mitosis and performs a final quality check to fix DNA errors, and also begins forming structures like spindle fibers for division. So spindle fibers go and grab onto the chromosomes and kind of just pull them apart, and then one side will take one side of the chromosomes and the other side will take the other side. And that's how you get the two cells, as it basically pulls the necessary stuff out and then it chops down the middle. Effectively, it doesn't actually chop, but that's the general idea. So let's talk about DNA and RNA info. So first you have to talk about DNA and RNA's pairings. So pairings are a big thing of DNA and RNA, and they have this little diagram, right? So what you see is A is connected with T and C is connected with G. A is adenine, T is thymine, C is cytosine, G is guanine, and A and T have 2 hydrogen bonds, and C and G have 3 hydrogen bonds. So RNA is slightly different. For the C and G section, it is exactly the same, with cytosine and guanine being paired up and having 3 hydrogen bonds. But for the A and T one, that is switched up. T is no longer there, T being thymine, and it is now A and U. So it's adenine and what's called uracil. And these also have 2 hydrogen bonds, so same amount. So now let's compare DNA and RNA just on a little table. So DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. I'll be talking a bit more how those things change it. So DNA uses thymine, as I brought up earlier, whereas RNA uses uracil. DNA stays in the nucleus and never leaves, but RNA leaves the nucleus, and I'll talk a bit about how that works. So DNA has this double helix shape, and the idea is it has 2 strands that kind of bend around. RNA just has a single strand, which is just one, and it has all its stuff going out like that. There's just one type of DNA, just DNA. But there's actually three types of RNA. There's mRNA, tRNA, and rRNA. So, as I brought up earlier, DNA is deoxyribonucleic acid, and RNA is ribonucleic acid. So this talks about its sugar, because the sugars on it matter a lot. DNA uses deoxyribose sugar, whereas RNA uses ribose sugar. And this just changes how it works, and this changes whether or not it's RNA or DNA. So DNA's purpose is to store genetic information, and it's the template for replication, whereas RNA carries genetic information from DNA to ribosomes for protein synthesis. So the way it works is DNA has its shape, right? And then one side of it gets pulled away, and RNA, say that this isn't the DNA anymore, RNA comes in, matches up with it, and makes a copy. So this is an opposite copy. Because let's say that you have A here for adenine, then you'd have uracil on the other side, and they connect like that, and you get a whole bunch of them. And this is mRNA, which is messenger RNA. So messenger RNA, once it does that, then leaves the nucleus, and the DNA is kind of pushed back together. So enzymes open it, and then I'm not sure how they go back together. And then, it disappears, right? And then the mRNA goes down to rRNA, and it kind of just like lies there. And then tRNA has what's called anticodons on it. And anticodons are three things. So again, there's adenine, cytosine, uracil, guanine. Yeah. So then it's a pair of three. It can be any combo of three. And if the opposite pair is on the mRNA, then it'll come down and connect to it, and the enzyme that it carries will just kind of go up and then be caught. And then, not enzyme, amino acid. The amino acid will go up, be caught, and then the rRNA just holds it all those amino acids, and then eventually it'll create a protein. So let's just give a little example. For example, if I had AAG, then the tRNA would be UUC. So that's mRNA, tRNA, and rRNA, and that's the different kinds. And eventually you'll get a little stop codon, which is so one of the tRNA will have nothing on it and come down, and then that'll basically mean, hey, we're done. We don't need any more stuff to come in. The protein's done. So let's talk a bit about cell processes, because each cell does its own little thing. So plant cells will do what's called photosynthesis. It's a very common idea. So it takes light energy, carbon dioxide, and water to create oxygen and glucose. So that's why when we breathe out carbon dioxide and it goes to plants, and then plants will be able to bring out oxygen. And that glucose it keeps, and that's how it's able to grow. So animal cells do this thing called cellular respiration. So it isn't just animal cell limited, and some plant cells do this too. But the idea is it's kind of the opposite way, right? They take glucose and oxygen, which came from the plants, and then they turn it into energy, which is sometimes called ATP, carbon dioxide, and water. So that can then be kind of filtered back into the plant cell with its light energy, carbon dioxide, and water. But yeah, it's a very simple idea like that. So let's talk about the different parts of DNA and RNA, right? So I brought up that adenine, thymine, cytosine, guanine, uracil thing. So this kind of goes a bit more in depth on those two, on that. So base pairs are a big part of the DNA. And the base pairs are a pair of nitrogenous bases in DNA or RNA that bond together, forming the rungs of the nucleic acid ladder. So as I brought up earlier, adenine goes with thymine, cytosine goes with guanine, uracil goes with adenine. And the idea is each one of those are the nitrogenous bases. And then when they combine, they're called a base pair. And you might question, how do they combine? They combine via what's called hydrogen bonds. Hydrogen bonds hold the complementary base pairs together. And again, the complementary base pairs, adenine, thymine, guanine, cytosine, uracil, adenine. So that's what's holding them together. And this is what I brought up earlier, where the guanine and cytosine have 3 bonds, whereas the adenine and thymine only have 2. So does the uracil and adenine. So let's talk about the nucleotide. And this is the basic building block of both DNA and RNA. Each nucleotide nucleotide. Oh, yeah. Each nucleotide consists of three parts: the phosphate group, the sugar molecule, and a nitrogenous base. So there's an image on screen right now, and there's the P for their phosphate, the S for sugar, and then it, in this image, holds in G. So G, again, guanine, it's one of the nitrogenous bases. Let's talk about the sugar phosphate backbone. So, as I brought up earlier, in the nucleotide, you have the phosphate, sugar, and then the G. So the sugar phosphate backbone holds just the phosphate and the sugar. And this is how everything is kind of held together. It's a structural framework of DNA and RNA, and it's made up of alternating sugar. So there's deoxyribose in DNA and ribose in RNA, which I brought up earlier. Those are just the different kinds of sugars that it uses. And there's phosphate groups that are covalently bonded together. Next is codon. So I brought this up earlier, but it's the three from the tRNA. So in the tRNA, it's anticodons, and then in the mRNA, it is codons, and then they connect, and then they drop off the amino acids. So let's talk about definitions of some important terms. A lot of these terms have C in them, so it's very easy to get mixed up. So this part is actually quite important. So first you have chromatin. Chromatin is proteins that attach to DNA, which condenses long DNA to help them fit in the nucleus. So it's a bit of an organizer. It'll take DNA, it'll wrap it around, but it'll wrap it around in a certain way where it's able to be accessed later on in the future if needed. And then this is basically the unwound form of chromosomes, which I'll talk about later. Next is the chromatid. So the first one was chromatin, the next one is chromatid. So there's one of two identical pairs of sister chromosomes. So it is the parts that make up the chromosome, and the two chromatids are joined by what's called a centromere, which I will talk about centromere later. And the chromatids are made of many chromatins. So that's how they're related. Chromatins will wrap around the DNA, and then it'll bring it, and then the chromatid is a whole bunch of chromatins, and then the chromosome contains all the DNA instructions necessary for cell function. So like I brought up earlier, this is where all the chromatins are put. So all the chromatins are put in the chromatid, and the chromatid is held by the chromosome. So the chromosome is made up of one or two chromatids. So it depends on which phase it's in. In G0, you'll only have one chromatid. But during S phase, you're creating your second one because you're ready for division. And then in G2, then it will have two, right? Because then it'll be pulled apart later on. And these are stored in the nucleus. Next is a centriole. The centriole is a cylinder organelle made of microtubules that help organize the mitotic spindle and aids in chromosome movement during cell division. So during mitosis is the phase where it kind of breaks apart. It will help basically pull apart the chromosomes and bring them to its respective sides. Next is centrosome. And this is not to be confused with the centromere, which I brought up earlier. And it's an organelle found in animal cells that serves as the main microtubule organizing center, playing a key role in cell division by helping to form mitotic spindles. It contains a pair of centrioles which assist in chromosome movement during mitosis and meiosis. So the centriole seems to be inside of the centrosome, and it's just so the centriole is part of the centrosome. Let's talk about the centromere. So this is the region of a chromosome that holds the two sister chromatids together and is the attachment site for spindle fibers during cell division. Next is the nucleus. So the nucleus, as I brought up earlier, is where all the chromosomes are stored. And chromosomes, again, as I brought up earlier, contain centromeres, chromatin, chromatid, and all that stuff. So the nucleus is the organelle in eukaryotic cells. So there's both eukaryotic and prokaryotic. Prokaryotic is the more simpler organisms such as bacteria, and eukaryotic is the bigger cells with more stuff inside of them. You'll see them in plant cells, animal cells, etc. So the organelle in eukaryotic cells that contain cell genetic materials in the form of DNA. It serves as the control center of the cell, regulating activities like growth, metabolism, protein synthesis, and cell division. Next is spindle fibers. So this is very related to both the centrosome and the centriole that I brought up earlier. So again, it's microtubular structures that form during mitosis and meiosis. And yes, there's a meiosis, I'll talk about that later. And they attach centromeres of chromosomes and pull the chromatids in mitosis, or homologous chromosomes in meiosis. So it's slightly different, the types of chromosomes and chromatids that are being pulled apart, depending on if it's mitosis or meiosis. But they're basically aiding and pulling apart during cell division. Let's talk about karyotypes. So karyotypes is the general appearance of the complete set of chromosomes in the cell of a species or in an individual organism, mainly including their sizes, numbers, and shapes. Karyotyping is the process by which a karyotype is discerned by determining the chromosome complement of an individual, including the number of chromosomes and any abnormalities. So you might see in this picture that one of them looks a bit off, and that is because those are the gametes, the X and Y's, and some parts of it, either the X or the Y, has codes for more DNA. So that's why that one is bigger, as the other one doesn't have to have as much stuff. Let's compare mitosis and meiosis. So mitosis and meiosis are both types of cell division. So mitosis produces two identical daughter cells. So you start out with one thing, and then afterwards you end up with two exactly same things, right? So the first cell is exactly the same as the second cell and the third cell. So from one cell, you create two identical cells. There's no difference between them. Whereas meiosis creates four genetically unique gametes, which are sperm or egg cells. So they're all like different than the starting cell, and there's a bit of a difference which I'll bring up later. Mitosis has one division, whereas meiosis has two. In meiosis, crossing over occurs in prophase 1, which increases genetic variation. So that's why it's four different ones instead of just four of the same. Whereas mitosis maintains the chromosome number, so diploid to diploid. So there's the exact same number of chromosomes, while meiosis halves it, going from diploid to haploid. So diploid basically means that there's two chromatids, which make up a chromosome, whereas haploid means there's just one. Meiosis is essential for sexual reproduction, while mitosis supports asexual reproduction and tissue maintenance. Both processes share very similar stages, with prophase, metaphase, anaphase, and telophase, but meiosis includes additional steps for genetic diversity. So it has a prophase 1, metaphase 1, anaphase 1, telophase 1, then a prophase 2, metaphase 2, anaphase 2, and telophase 2. So there's much variation between them all. Yeah, that's my quick 17-minute recap. That's not quick. But if you want to get up scuff on about grade 9 or grade 10 biology, that's how you do it.